So how did we specifically address this challenge? Well, what we did is we combined the best of both worlds by using optical imaging, so being able to advance your 2D planar imaging, and then moving it to a 3D format. And so in this case, we add in tomography without the use of radioisotopes. We add in depth information that you get by acquiring data around the animal. You add in quantification as well, as we'll see later on in examples where we have a reconstructed signal that can then correlate to a specific concentration. And finally, this allows for the best pathway to a clinical translation, as it is a 3D tomographic method. So in order to do this, we looked at the near infrared region and specifically designed our instrument to really highlight this. And so NIR has optical light tissue interaction, as biological tissues are highly diffuse medium. And so you have both absorption and scattering. And in the near infrared window, we have the so-called biological window. So you can see in this graph on the left, as we increase the wavelength, the absorption dramatically decreases from hemoglobin and oxyhemoglobin, which are two components found inside biological tissue that are strongly absorbing. And then in this window, scattering becomes highly dominant. And so also, because we have least amount of absorption, we can also get the best depth penetration, as these longer wavelengths of light are able to penetrate deeper without being absorbed or as much. And so using these near-infrared fluorescent probes or infrared fluorescent proteins or nanoparticles, we can then use this in vivo optical imaging and get the best depth information along with the least amount of absorption. So, as I mentioned, I'm now about to introduce what our optical tomography technology is, and that stands for FLECT. FLECT is fluorescence emission computed tomography. And here on the graph on the left, we can see a nice animation. Before it starts, I'll quickly go through it and say that the black parts are the photodiode detectors that we use. We have 48 in a ring formation. And then we also have a laser light source. And the red blocks represent the filters that are put in front of the detectors. So as we rotate, because all these components are all placed onto the same rotating gantry, as we rotate around the animal, we have different source points that illuminate the fluorescence or fluorescent agent inside the animal. And so we're able to collect both epi and transillumination fluorescence at the same time. We use lasers for maximum fluorescence efficiency. And in one slice, we can collect upwards of 116 source points, meaning 116 times 48. And then we move in 1 millimeter increments transversely along the axis of the animal. And so, as you can imagine, these data sets become quite large as we acquire the emitted fluorescence signal. In addition to this, the fluorescence emission computed tomography is also mated to a CT gantry. So we take these two tomographic imaging modalities in sequence, one and then the other, and then we use the CT information to inform the reconstruction algorithm and put this into a 3D volumetric format that can then be visualized. So as we move on to explaining how the tomographic reconstruction works, we use a 3D finite difference model of light propagation, and this accounts for different optical properties based on the different tissue types inside the animal, related to both absorption and reduced scattering. And then we also, like I mentioned, use the CT to inform and enhance the reconstruction input parameters. So we create a complex mesh grid after these single slice acquisitions and a volume is created. Then this mesh grid is then input with these specific optical properties. And then finally, we throw it into the engine and it is able to spit out a nice DICOM format, which can then be visualized by VivoQuant, ImageJ, or any imaging suite program that you like. And depending on the location of the animal, the optical properties present, whether it be lungs, tissue, liver, the brain, etc., these really do change quite a bit and can change how the reconstructed signal gets visualized. So it's really important to use the CT to inform this reconstruction engine. And so, yeah, like I mentioned before too, the main point is to optimize and fully utilize your optical imaging study to the best of its extent by moving from a 2D planar to a 3D tomographic method. And so where you lack in localization and depth information in your 2D images, now you can get that with a 3D tomographic visualization, in addition to a nice anatomic reference that tells you where your molecular agent is actually being reconstructed inside the animal. And so some of these images I'm sure you're familiar with, including the strictly 2D normal photographs, followed by the overlay of the molecular agent on top, and then different parts of the body. Then moving to a limited angle 3D tomosynthesis, which can project it onto the surface. And then finally, to our method, our 3D fluorescence tomography, where we use 360 degrees of data acquisition and then reconstruct it within the animal. So our instrument here, it can be visualized in that nice picture. It's a benchtop system. As long as you have a sturdy enough table to support the weight, it can fit into a regular size lab. And the lasers that we use are 4 standard lasers, 3 used for fluorescence excitation and 1 for creating the surface boundary. We have associated fluorescence filters in the centered around 700, 800, and 850 nanometers with the associated full width half max. And then we can confidently say that we can image a sensitivity of 1 micromolar, but in practice we've seen less than that. So it really depends on the application and how much light you can get out. In addition to our X-ray CT, we also have the addition of these nice X-ray filters, which can reduce beam hardening effects and can also enhance soft tissue contrast and other applications like bone imaging that really make the CT anatomic data stand out. And so the data that gets output from these data acquisitions can be seen here. And I'll start from left to right. We'll start with the maximum intensity projection, which is a nice rotating GIF around the center axis. And then we have the corresponding sagittal, coronal, and transverse views that we'll show in the new or these next application areas. And as you can see, we have a nice fly through this whole image stack. So as you can tell, there are quite a few, on the order of hundreds, of individual images that get generated as a result of acquiring data in 360 degrees. So now I'll hand it back over to Austin, who will talk about the specific application areas that our customers have been investigating, and also why it's important for them to choose, why they chose FLECT, and how it helped them inform the research.
Leave you with one thought exercise, please, which is now that you know that 3D optical imaging can see deep within the mouse for in vivo work, how can we help your research? How can we help move your work and move that work forward and help to cure some of the diseases that we're all here to tackle?